It is so shocking to know that India stands at the third position globally for the highest number of cancer cases, as well as the third position for breast cancer. It is not something to be proud of. Every eighth person out of 10 people are either suffering from cancer or at the risk of cancer due to hereditary or lifestyle factors. So, there are several methods nowadays to detect the presence of cancer, the presence of tumors, to be accurate, like mammography, biopsies, sonography, MRI, blood tests, PET scans, also to verify how much the tumor has spread into a body. But what if there was a way by which we could predict if the person has a chance of developing cancer? So, due to advances in medical sciences, a diagnostic technique called FRET is used for detecting mutations in genes of people who have a history of cancer. So, hello, I am Sanika Ranadive, student of MSc Biotechnology Part 1, pursuing from Pillai College of Arts, Commerce and Science. And today I'll be focusing on this topic through the paper, a unique threat approach towards detection of single base mismatched DNA in BRCA1 gene from the journal Material Science and Engineering C, um, which was published in April 2019 by Elsevier. Um, so let's move ahead. So before exploring into what actually is there in the paper, how about a short intro to these terminologies of FRET and BRCA gene? So FRET, or also termed as Foster or fluorescence resonance energy transfer, which is abbreviated as FRET. So it's a type of detection system which involves use of fluorophores in close proximity, a donor and an acceptor. So the fluorophores could be nanomaterials like the quantum dots or abbreviated as QDs and silver nanoparticles or known as AGNCs. So it was introduced by Theodore Foster, so that's the name, and it was later modified and experimentally confirmed by Stryer and Hoglet. Next is breast cancer. So the BRCA gene, which is present on the chromosome 13 and chromosome 17. So these are tumor suppressor genes, which means that they repair any DNA damage, or if any tumor has been formed, they try eliminate it before it becomes a cancerous tumor. So one copy is inherited from each parent, and an individual with a defective copy with one defective copy can survive on the other, but due to today's lifestyle conditions, due to today's way of eating habits, living habits, sleeping cycles, the second copy, the healthy copy, also tends to mutate, and at that point the cancer cannot be controlled. So what now, coming back to the topic, what the researchers actually wanted to do so they wanted to detect the presence of that gene in the patient with high chances of who has high chances of developing cancer in the coming future. So for this they chose to detect the SNPs, which are present every 1,000 base pairs, as well as the single match base pairs. So the quantum dots were bound to the cytosine thymidine abundant ends of the DNA, which became the donors, and the silver nanoclusters, the AGNCs, as the acceptors, which detected the fluorescence. So for any experiment to be conducted, it cannot be done without the essentials. So, moving to the requirements. So for the apparatus, it was instrumentation was done by where Perkins Elmer Lambda 25 spectrophotometer, and fluorescence measurements with xenon lamp for excitations were used. Along with that, transmission electron microscope, of course, to detect the nanoparticles. So on the other hand, the DNA samples were acquired by polyacrylamide gel electrophoresis, or abbreviated as PAGE, and Then the DNA regions, that is the wild type and or the normal DNA, the mutant DNA, a non-complementary target, and a DNA was labeled by the silver nanoclusters. The silver nanoclusters and quantum dots were prepared freshly using analytical grade chemicals purchased from Merck. So, what exactly was done in this experiment? Okay, so moving ahead to the methodology. Initially, what we have seen, the SNP regions of the BRCA1 gene was isolated by page. Then the silver nanoclusters and the DNA fluorescent probe were prepared, as well as the CDTE quantum dots were synthesized. Then it was labeled by another set of silver nanoclusters to the target gene, and all these sequences were processed through agarose gel electrophoresis at 60 volts for 45 minutes. The QD insertion into the DNA caused a change in the electrophoretic behavior, and the age results were observed by UV transilluminator at 312 nanometer. So, in order to determine the hybridization effect of complementary and non-complementary uh, complementary, that is the single base mismatch, targets on the fluorescence intensity, different concentrations of the or target single base mismatch DNA in BRCA1 gene were added to obtain the DNA AGNC solution. The hybridization was carried out by incubation for 1 hour at 37 degrees Celsius. And then the fluorescence was measured, which was performed at room temperature. So, coming to the crux of this experiment, the results, which are over here, listed out over here. So the first thing that was noticed was between the quantum dots and the silver nanoparticles. Both overlapped at the same wavelength range, that is 430 to 530 nanometer. Then the morphology of the silver nanoclusters was determined by transmission electron microscopy, indicating 2 nanometer diameter of the nanoclusters present between the DNA scaffolds. So the desired size, then the next was the desired size of the quantum dots is 2 nanometer, that was viewed as a blue-green fluorescence. So moving ahead, how did the QD and the DNA interaction was affected, was observed and noted. So it was seen that the wild type DNA generated a significant signal, whereas the QD in single base mismatch did not. So it was much of a moderate signal. Along with this, the normal type and mutant type of DNA generated quantifiable signals, detectable signals. So thus it could be determined to then the analytical performance was examined. So how it was done is to examine the analytical performance of the designed method for detecting the complementary and the non-complementary target. So the fluorescence spectra with the non-complementary target and the fluorescence spectra with an excitation of 370 nanometer. So were recorded after 20 nanomolar of the CDTEQDs was added in the detection solution containing DNA AGNCs and the target sample. So when there was an increase in the mutant target concentration from 1.5 into 10 raised to 10 molar, uh, 10 raised to 10, minus 10 molar, to 1.5 into 10 raised to minus 6 molar. So the fluorescence intensity of the quantum dot silver nano clusters with excitation at 370 nanometer was showed with a linear increase. So here is the graphical representations and diagrams or images as shown. So the first shows the complex, it's a schematic version of the QD's attachment to the DNA. Then it's the UV visible and fluorescence emission spectrum. Then there's the gel electrophoresis, the results obtained of the gel electrophoresis. Then these are the fluorescence emission spectra. Also the effect of pH. Now finally, in the conclusion, with this research, there was a big breakthrough in medical sciences that SNP sequences can be detected by signals emitted as a diagnosis for mutated genes, and FRET being one of the reliable technique. Uh, and because FRET was a reliable quantitatively quantification method for the target concentration, as well as it could quantitatively differentiate between the wild type and the mutant type DNA sequences. So to end with my presentation, these are the references used. The first one being the paper from which this entire presentation is being prepared, and the next five are the top five references used by the authors from whom I have referred this paper. Um, and thank you for your time, and I hope you find this informative.